Hi everyone, welcome back. Wow, we're already halfway through the term. So what we're going to be doing this week is we're going to get into cellular respiration, which can be tricky for some students. So we're going to work our way through it one step at a time. So hopefully by the end, you'll have everything down, okay? There's quite a bit of learning outcomes this week. So we're going to talk about redox reactions, which we addressed last week. So just kind of a brief overview of that. And then we're going to trace the pathway of glucose oxidation during a process known as glycolysis. And then we'll see how that oxidation of pyruvate is further oxidized in the citric acid or Krebs cycle. And then we'll talk about the steps of the oxidative phosphorylation, where we have the majority of the ATP being produced per glucose molecule. And then we'll talk about the two main forms of fermentation, one that can occur inside our own cells and one that some microbes like yeast use in order to get their energy. So we are in chapter 7 this week. We will touch on a little bit of chapter 9 at the end of the second lecture. Okay. So with chapter 7, we've already talked about catabolism. So that was basically what we talked about at the end of last week. So do make sure you know what a redox reaction is. Remember the oil rig, or however you want to remember it. So oxidation is losing electrons. Reduction is gaining electrons. Okay, so that's why I always use that acronym oil rig. So oil, oxidation is losing, reduction, rig is reduction is gaining. So do make sure you know that. And then catabolism versus anabolism. Okay, so we're going to actually start out just kind of reviewing what cellular respiration is. Again, we're going to do an overview of it, and then we'll get into each step specifically. Okay. So let's start out by doing the overview. So with aerobic cellular respiration, it occurs in 3 main stages. There's a couple in between stages that we'll talk about as well. So we usually divide it into 3 stages. We have glycolysis, we have the citric acid or Krebs cycle, and oxidative phosphorylation, which is where the electron transport chain is. So glycolysis is the first step of cellular respiration. And it actually occurs outside of the mitochondria in the cytoplasm or in the cytosol. And that's where the molecule glucose will start to be broken down. Okay, so as it's being broken down, it will be broken into basically 2 3-carbon molecules, which we call pyruvate. So through the end of glycolysis, we actually do gain some ATP through that process. And then from there, the pyruvate is going to enter the mitochondria, where it will be oxidized further and release more electrons. Because remember, oxidation, you're losing electrons. So it's going to release more electrons, which will be used to attach to that NAD plus to make it NADH, as well as the other electron carrier FADH2, and carry those electrons further in the inner membrane of the mitochondria. So during that process, that's what we call the citric acid or Krebs cycle, and it is a cycle. And then finally, we will take all of those electrons that the electron carriers have transported, and they will be used through an electron transport chain to create this proton gradient. And this will cause, cause the formation of ATP through an enzyme called ATP synthase. And that's where the majority of ATP synthesis occurs, is in this last stage. And that's going to occur in the inner membrane of the mitochondria. So let's start out with a video, again, as an overview, and then we'll go through the different steps specifically.
As this mountain biker heads up the trail, The breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, one carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Okay, so again, I mean, we saw that video last week, but I did want to reintroduce it to get us started in this complicated process. And like I mentioned last week, in your textbook, things are color-coded. So when you see kind of that bluish color, that indicates glycolysis. The orange color would be the citric acid cycle, and then the purple is the electron transport chain, okay? When we go through, you'll see these figures and you'll notice, okay, so here we have the first step, which we have the mitochondria. Here's the giant mitochondria. And you can see that glycolysis is occurring outside of the mitochondrium. So these figures that you see in your textbook are actually quite useful because it'll go through what goes in and what comes out of each stage. So this is something that might be helpful to maybe create a chart where you have the different parts of cellular respiration and then include what's going in and what's coming out of each stage. So like the inputs and the outputs. And then that way you can keep track of some of these molecules as they're changing and reforming into new things. Okay, so again, we're just doing an overview. So we're going to start out with glycolysis. And again, you notice it's occurring outside of the mitochondrium in the cytoplasm or cytosol of the cell. So this is where that nutrient glucose comes into play. So glucose, if we recall that C6H12O6, that chemical formula, and that's going to be oxidized. It's going to be broken down into pyruvate, which is that 3-carbon molecule. So one glucose will produce 2 pyruvates. And during that time, we're going to obtain a net positive of 2 ATP molecules. So just by breaking down glucose into pyruvate, we will gain 2 ATPs. Okay, we will also gain some electron carriers. Okay, so we will transfer, the glucose will undergo oxidation, so it'll lose electrons, which will then be forming into NADH. Okay, so we have a couple electron carriers being produced. Okay. And then from there, the pyruvate will enter the mitochondrium of the cell, and it will undergo some further oxidation to form something called acetyl-CoA, and we'll talk more about that. And so during acetyl-CoA, there's no ATP being produced. It's just transferring the pyruvate into something else that will then go into the citric acid cycle. In the citric acid cycle, we saw from the video, the pyruvate or the acetyl-CoA is going to basically combine with a 4-carbon chain already, and it's going to undergo this transformation, this cycle where it's going to further be oxidized and release more electrons, and it will eventually allow us to form ATP. Okay. And then finally, the bulk of that, the ATP synthesis will occur in this last stage of our cellular respiration. So all of those electron carriers that we have gained from glycolysis and the citric acid cycle will then be carrying their electrons to the electron transport chain. So that electron transport chain is going to allow for the electrons to transfer through the inner membrane of or throughout the proteins in the inner membrane of the mitochondrium. And by doing so, it's going to create this gradient where the protons will get pumped on the other side of the inner membrane of the mitochondrium. So then during this process, we're gaining basically this positive charge on the other side of the inner membrane. And so all these protons, all these hydrogen ions are kind of situated at a high concentration. Finally, that will allow for, as the proton channel opens up and the ATP synthase will start synthesizing those protons, that will use that energy to combine that ADP and the phosphorus together to form ATP molecules. And finally, we're going to end up with a bunch of electrons that will then be attached to oxygen in order to form H2O. Okay, so we'll go through it and just realize that there is about 3 steps to this process. The majority of the ATP is created at the end, okay, about 90% of the ATP. All right, so let's get into the details. So we're going to start out with glycolysis. So glycolysis, glyco, that's kind of sugary, right? And then lysis, think splitting. So it stands for sugar splitting. We're taking that sugar molecule, the glucose, that monosaccharide, and we're going to break it down into 2 molecules of pyruvate. So you can see in the simplistic form here, we have glucose, C6H12O6, so it has 6 carbon atoms. That's going to be our input. And then because we are taking a polymer and we're breaking it down into the 3 carbon chained molecules, we do need some energy to get this going. Okay, so we will be using what's called the energy investment phase of glycolysis, where we're actually going to need 2 ATP in order to power the breaking of the glucose molecule. Okay, so glucose needs to be broken down, and it can't do so on its own. It needs an investment of ATP to be able to do that. So ATP, 2 ATP will be used, so that energy will be used to break that glucose molecule into our pyruvate. And so you notice with the pyruvate, we have the 3 carbon chain there. And so that 3 carbon chain is going to, basically be migrating further until we end up with something called pyruvic acid. So you can see where that ATP comes into play. So ATP is going to undergo its own reaction, okay, so where it's going to basically go from ATP to ADP with that little extra phosphate group attached to the pyruvate. Okay. And then during this process, in addition to requiring 2 ATP, we will continue doing the stripping of this pyruvate, where it's going to become oxidized. Remember, oxidation, we're going to lose electrons. So it'll lose electrons and create 2 NADH from that. So remember, NADH, that's that energy carrier. So it goes from NAD plus, and then with the addition of the electrons, becomes NADH. So in this step, you can see what's happening. We've used 2 ATP, and we've gained 2 NADH, okay? We further undergo the process to form pyruvic acid, and in doing so, we create 2 more ATP per molecule, okay? So at the end, if we look at the input, we have glucose. We've used 2 ATP, but we've gained 2 plus 2, 4 ATP. Okay, so we've gained 4 ATP, but we had that initial 2 investment. So our net gain of ATP, 4 minus 2 is 2. Okay, so at the end of glycolysis, we've gained 2 total ATP. Okay, we got to pay off the investment first. We've also gained 2 NADH, and then we've gained 2 pyruvic acids. Okay, so our input was glucose, our output was 2 pyruvic acid and 2 ATP and 2 NADH. All right, so just make sure, again, making a chart will be helpful. So you write glycolysis, and then for the inputs, you have glucose. For the outputs, you have 2 ATP, 2 NADH, and 2 pyruvic acids. Okay, and all of this is occurring where? In the cytoplasm of the cell. So that's the first stage, and remember the first stage is occurring outside of that mitochondrium. And so the NADH, those electron carriers, are going to be traveling into the mitochondrium, and the pyruvic acids will be moving into the mitochondrium in this next step. Okay. So here's just an overview. Some of you, it might be easier to see from this viewpoint, the reaction. So we have our energy investment phase. So the glucose, in order for the glucose to be broken down, we need to use 2 ATP. So the ATP will basically get used and end up being formed into 2 ADP plus 2 phosphate. Okay. And then from there, the two ADP and the two phosphates will go ahead and be used in the energy payoff phase, where we're going to end up with 4 ATP being formed. And we're also going to end up, by stripping those electrons, we're going to end up with 2 NADH. Okay. You can also see there are a couple of hydrogen ions that were formed. So that gives us our 2-pyruvate. And then, so that's our net. We have glucose with the 2-pyruvate. We have 4 ATP being formed, but again, we have the energy investment phase to account for. So we have a total net of 2 ATP. And then we've gained 2 NADH electron carriers. Okay, so those are the payoff phase. And then again, here's another way if you want to look at it in an even different way, where we have the structural formula. So here's our glucose. It's going to be broken down, okay, with the conversion of ATP to ADP. So that will be broken. Again, you don't need to memorize any of that, but we're going to use 2 ATP to break apart that glucose molecule until we finally have our eventually the 3 carbon chains. Okay, you don't need to memorize any of these terms. Okay, just realize that glucose, in order to form these 3 carbon chains, we need 2 ATP molecules. So this is just showing where the 2 ATP molecules are being used. Okay, and then from there, here's one of our 3 carbon chain. Okay, again, you notice I'm not even using the proper term. I'm just using 3 carbon chain because you don't need to know the chemical term. And that will be further oxidized, releasing those electrons to form NADH. Okay, and in the process, we'll be gaining 4 ATP. Now, finally, we have our end result, our pyruvate. So you can see how pyruvate, what it looks like. Okay, you don't need to memorize the structural formula in any way, but you can see how that pyruvate is going to then, there's 2 of them, be processed further, stripping away more electrons during the process of the citric acid cycle. Okay. So here's a question for you. For each glucose molecule process, what are the net molecular products of glycolysis? All right, so what did we end up getting? We ended up getting 2 pyruvates, okay, and 2 ATP and 2 NADH, okay? You don't need to worry about the protons, they just get used. But this is basically the input, and then you see that we have different enzymes. You don't need to memorize the enzymes, plus that 2 ATP energy investment pays off for a net total of 2 ATP plus 2 pyruvates, plus I guess I should write 2 NADH. All right. So now we've oxidized glucose, right? We've broken it down, we've stripped some electrons from it, and so now we're going to proceed into the next stage, okay? So the next stage, we are going to further oxidize pyruvate and complete the citric acid cycle. And so this stage, there's actually a first part of it before we actually get into the citric acid cycle. So we're going to talk about that one first, though some textbooks just lump them together, but you know, you can just separate them as well. So I've kind of done both. So we have, this is part of the citric acid cycle, but this takes place outside of the cycle part of the citric acid cycle. And this is where that pyruvate or pyruvic acid is going to undergo oxidation. Okay, so what does oxidation mean? Oil rig, it's going to lose more electrons. So we're going to end up with that pyruvic acid. It's going to release one of those carbons. Okay, so as it's releasing one of those carbons, that carbon will become carbon dioxide. Okay, so we'll be releasing some carbon dioxide in the first step. And then that leaves us with 2 carbons, which forms this acetic acid. This acetic acid will combine with a coenzyme A to form acetyl-CoA. So this is called acetyl-CoA because it has acetic acid and it has coenzyme A. All right. During this process, though, as it's forming acetyl-CoA, we're going to have the stripping of some electrons. So that will form our NADH, our electron carrier. Okay, so this kind of in-between phase, we've taken our pyruvic acid and we've stripped 1 carbon from it to form carbon dioxide. And then we've stripped some electrons to form an NADH. And our final output is this acetyl-CoA. Okay. But remember, how many pyruvic acids do we start with? With 1 glucose molecule. So that glucose molecule produced 2 of them. So this is done twice. Okay, so the output here would be 2 acetyl-CoAs and 2 NADHs. And then from there, we have, this is just showing, here's the cytoplasm, here's our 3 carbon pyruvate making its way into the mitochondrion, and we have pyruvate oxidation taking place where we're going to get that electron carrier taken care of, 2 of them because we have 2 of these, and then the formation of acetyl-CoA. All right, so that's pyruvate oxidation. All right, so here's a question for you. Which molecule in the figure below has been reduced? What's that term reduced mean? Oil rig. So which one has gained electrons? The NAD plus has gained electrons, right, to form NADH. So that means that NAD plus was reduced. Remember that weird thing where you're gaining electrons, but you're being reduced, okay? So just remember the vocabulary there. So NAD plus is reduced to NADH because it's gaining the electrons. Okay, pyruvic acid is being oxidized because it's losing the electrons. All right, so now we have our acetyl-CoA, we're going to undergo the citric acid or Krebs cycle. So sometimes you'll see citric acid cycle, sometimes you'll see Krebs cycle. They're the same thing, just different names. I can't remember, I think the citric acid cycle is the more used term these days. In the past it was more Krebs cycle. I always learned it as the Krebs cycle. So I'll use both names interchangeably, okay? But either way, they're the same thing. So this is taking place in the mitochondrium, and we're completely breaking down the pyruvate to create carbon dioxide. So that's one of the byproducts of cellular respiration, right? We're taking glucose and oxygen, we're creating carbon dioxide, water, and ATP. So you can see this acetic acid, well, it's the acetyl-CoA, the CoA part, the coenzyme has broken off, and then basically this acetic acid is going to join this 4-carbon chain that's already part of the citric acid cycle. And then in this process, we now have a 6-carbon chain again, and this citric acid cycle is a series of events that take place, or a series of reactions that take place, where we systematically remove the carbons to produce the carbon dioxide and stripping it, so we're oxidizing this to form a lot of electron carriers. And we do have a we actually produce 2 ATP in the process. Okay. So here's the input, acetic acid. Here's the output. We've gained 2 carbon dioxides, 1 ATP, excuse me, 3 NADH, and 1 FADH2, just from 1 acetic acid. Okay, so we have multiply that by, remember you got 2 of them, so that would be 4 carbon dioxide, 2 ATP, 6 NADH, and 2 FADH2 total, okay, from that original glucose molecule. All right, so let's take a look. Okay, now the citric acid cycle, it does have a series of steps, and you don't need to memorize any of those steps. So I like this particular figure. I'll show you a more complicated figure in a minute, but this particular figure is all you need to know. So we're taking in that acetyl-CoA, okay, remember the acetic acid plus the coenzyme A. That coenzyme A gets recycled, okay, so that's going to go pick up another one. So now this acetic acid will enter the citric acid cycle, where it will attach to another 4-carbon chain, which will be used. And these 2 carbons that were invested in the citric acid cycle will eventually get stripped to form 2 carbon dioxides. So you see the 2 carbons, and now you have 2 carbon dioxides. Okay. In the process, we're going to be stripping electrons. And by stripping electrons, we're going to form 3 NADH and 2 ATP and 1 FADH2, okay, per acetyl-CoA, or the initial one. Okay, so here's the complicated part, only for those that are really interested in learning it. You don't, I've never memorized, well, I probably did at one point, but it's got to be 20 years last time I memorized all the different parts of it. So you certainly don't need to either. But just watch what's happening with the carbons, okay? So here we have our acetyl-CoA, so there's the 2 carbons. Remember, the CoA gets recycled. We have our 2 carbons enter the cycle. And they're going to attach to this 4-carbon chain. Don't even need to memorize that. Okay, so now we have a 6-carbon chain. Ignore the name. Okay, so now that 6-carbon chain is going to be oxidized. It's going to go through a process of being oxidized. So during that oxidation process, it's going to be stripped of electrons and dropping off the carbons as they go. Okay, so you can see our 6 carbons now become 5 carbons with 1 carbon dioxide being left. Our 5-carbon chain becomes 4-carbon chain with 1 carbon dioxide lost. Okay, during that process, you're noticing we're gaining electrons in our electron carriers, and we even gain an ATP. Okay, and then you can see this 4-carbon chain, ignore the name, will further get oxidized, strip some more electrons. Okay, and then finally, we're going to end up with this 4-carbon chain, which will then get stripped some more until we end up with that original 4-carbon thing that was used at the beginning. So we're adding these 2 carbons, and then those 2 carbons eventually leave as carbon dioxide. Okay, so you don't need to really identify the names of the different carbon chains from there. So just realize you start with 2, and 2 of them become byproducts, okay, as carbon dioxide. All right. So here is the summary. So in the citric acid cycle, we have 2 acetyl-CoAs go in, and then we end up with carbon dioxide, well, 2 of them. I don't know why I didn't have 2 there. So 2 carbon dioxides, 2 ATP, 6 NADH, again we start with 2, so double everything, 6 NADH and 2 FADH2. So in your chart, you have your acetic-CoA, there's 2 of them. You end up with 2 carbon dioxide, 2 ATP, 6 NADH, and 2 FADH2s. Okay, so basically we've netted an extra couple ATP plus a bunch of electron carriers. Okay, so this is the purpose of the citric acid cycle, is mainly to get those electron carriers. So here's our summary so far. In glycolysis, we've gained 2 ATP net. Citric acid cycle, we've gained 2 ATP net. But where do we get all the rest? Okay, there's like 32 that get produced total. So all the rest are going to be occurring in the third stage. So here's a checkpoint question. What is the total number of NADH molecules generated during the complete breakdown of one glucose molecule to 6 molecules of CO2? Hint, combine the outputs of glycolysis, pyruvic oxidation, and the citric acid cycle. Okay, so how many NADHs did we gain through glycolysis? We gained 2, right? How many NADHs did we gain from pyruvate oxidation? 2, right? So far 2 plus 2 is 4. And then how many did we gain from the citric acid cycle? 6, okay? So we've gained a total at this point of 10 NADHs, which are filled with electrons, okay? A couple electrons each. So that's a lot of electrons that are going to be transported into the inner membrane of the mitochondrium for the next step. The final step, oxidative phosphorylation, okay? So in this stage, we're taking those electrons that we've gained from the previous stages, and we're going to make a bunch of ATP. So this oxidative phosphorylation, so this is going to be basically 2 steps. So we're going to have something called the electron transport chain, and then we're going to have something called chemiosmosis. So in this step, we're going to see how those electrons that have been carried by those electron carriers, so they've been, here's the electrons, they were shuttled in by those electron carriers. There's the NADH, also FADH2. They're going to finally release those electrons, okay? So these electrons get carried across basically the membrane of the electron transport chain here. And so during this process, this is kind of an energy staircase where we have the electrons flowing through. And as the electrons are flowing through, that's going to pull hydrogen across from the inner membrane to just outside of the inner membrane. Okay, so remember let me go back. Remember this mitochondrium, it's got these little folds in it. Okay, so the inner membrane is where oxidative phosphorylation takes place. So we're going to kind of pump some protons across these little cristae, the inner folds. All right, so you can see how the electrons are traveling down the energy staircase, and who's waiting for them at the end? Oxygen. So this is where oxygen is finally coming into the equation. So that oxygen we breathe in is waiting patiently to gain those electrons. Because remember, oxygen likes electrons, right? It needs 2 to fill its its valence shell. And so it's going to accept those electrons at the end of the electron staircase. Okay, and then some of the extra protons will combine, or hydrogens will combine with that oxygen to form water. So you can see this electron staircase, we're pumping those electrons across, and they're being received by the oxygen, and then finally we end up with water as a byproduct. Meanwhile, we got all those protons ending up on the other side of the membrane. So we end up with this proton gradient, or hydrogen ion concentration gradient. And so that's going to cause a flow of hydrogen from greater concentration to lesser concentration, right? So it's going to go down the gradient. And while it's going down the gradient, as they flow through this, this is called ATP synthase, this enzyme protein complex, this is going to allow for enough energy to combine ADP plus phosphorus to form ATP, which is the moneymaker. This is what we want. This is the end result. Okay, so this is where all the hydrogen comes through, funneling through, allowing for enough energy to combine ADP plus phosphorus. Okay, so let's take a closer look with those terms that we were talking about. All right, so we have our carriers. Remember our electron carriers, they alternate from reduced and oxidized state as they accept and donate electrons. So remember throughout the time we had NAD plus being reduced, gaining electrons to form NADH. Okay, and then same with the FAD, it's going to be gaining electrons to form that FADH2. So those electrons, finally that NADH will be reduced to NAD plus as the electrons are stripped off to the electron transport chain. The FADH will get reduced to FAD and be stripped of the electrons. FADH. So FADH2. Now that those electrons have been stripped, they're going to pass through the proteins that are embedded in that inner membrane. And those proteins, they include something called a cytochrome, which has an iron atom on it. So that's going to allow that transfer of electrons through, basically. And then finally, at the end of the electron transport chain, we have oxygen as the final acceptor of those electrons. So O2, oxygen molecule O2, is going to be the electron acceptor. It's going to end up accepting those electrons as they drop through. So the electrons drop in free energy as they go, and they're finally going to pass to the electron acceptor, oxygen, okay, to form H2O. All right. So let's take a closer look with the figure here. So we have our NADH becomes reduced. So those electrons get stripped, and then it's forming NAD plus as the, just from being reduced. And then you can see these protons embedded in the inner membrane here. The proteins, excuse me, form this electron transport chain. So the electrons are going to be carried across this with free energy until finally they get picked up by the oxygen molecule at the end to form water. Okay. And then now we have, you see that the hydrogen ions, they get pumped across the membrane. Okay, so they're going to be pumped across during this process, during the electron transport chain, to form a concentration gradient. So we're going to end up with a lot of hydrogen on one side of the membrane, not so much on the other side. So this is where the harvesting of energy occurs. So as the hydrogen ions pass through the little channel there, they're going to allow for a bunch of ATP generated from the ADP and phosphorus. All right, so we're harvesting the ATP energy. So basically what went in, we had a lot of electrons, and what came out, we had water and ATP. Okay. So here's just a closer look at the electron transport chain. Don't memorize any of those protein complexes, okay? So you don't need to know any of that, but just realize that here we have that stripping of the electrons. The NADH gets reduced. So there goes the electrons. So the electrons are in this yellow arrow, okay? And then now we just have NAD+. So see how those electrons are traveling through basically the membrane here. There's that cytochrome, see that contains that iron, and then you can see it finally, there goes the electrons. They're going to be accepted by that oxygen and eventually form into the H2O. Right. So here's the question, what would happen if there was no oxygen? Okay, so let's say we don't have this at the end. So ignore that. What happened? Uh-oh. Well, in this case, there's no oxygen that are pulling these electrons through. So the oxygen's accepting the electrons, so it's trying to get those electrons closer to it. So it's essentially pulling it through the transport chain. So without oxygen, then we don't have an electron transport chain. Okay, so this whole process stops then. And so we don't, the NADH just ends up not being able to strip its electrons, and then we don't produce any ATP as the product. All right, so we've pretty much talked about the electron part, the electron transport chain, but what about those protons? So let's get into a little bit more detail about that. So we know that the electrons transferring through the electron transport chain causes proteins to pump the hydrogen ions from the inner membrane complex matrix to the, or excuse me, from the inner mitochondrial matrix into the inner membrane space. So now we have a high concentration of that hydrogen ions in this inner membrane space. Okay, it's kind of outside of the inner membrane. So now we need to, this creates a gradient, right? So we have all of these hydrogen ions on one side of the membrane. And so we're going to use those hydrogen ions. They're going to move across the membrane, passing through this ATP synthase. So ATP synthase is that protein that's embedded in the inner membrane, and it's got an ACE at the end, so it's an enzyme. And it's going to basically synthesize ATP. That's its job. It's synthesizing ATP with that with the proton channeling through. So the proton motive force is what we call it, as those hydrogen ions are channeling through the ATP synthase. So that ATP synthase uses all of that exergonic flow of all of those hydrogen ions to drive the phosphorylation of ATP from ADP and that phosphate. So chemoosmosis uses that energy in the hydrogen ion gradient to drive cellular work. Okay, so think of chemoosmosis, so that's basically that chemical gradient from the hydrogen is going to be used to drive that cellular work, the creation of ATP. So let's look at a visual, make sure we understand it. So here is, remember, here's our membrane, phospholipid bilayer, and then here is that ATP synthase, that protein that's embedded in the inner membrane. Okay, and you can see it's a pretty complicated looking thing. Okay. Schematic drawing of it, but we have all of this huge hydrogen ion concentration on the other side, okay, in the inner membrane space. Here's the matrix inside, okay, so these hydrogen ions are going to want to go from high concentration to low concentration, right? So they're going to be going through this ATP synthase enzyme, and as they're doing that, okay, this is creating an exergonic reaction. So all of those hydrogen ions are going to allow for ADP and phosphate to join together to form ATP. So there's enough energy through this process to drive this ADP plus phosphate to form ATP reaction. So we're just going to be pumping all of these ATP molecules are being produced. Okay, so they're getting synthesized as this enzyme is working its magic, driving the chemical reaction. Okay, so here's the two processes together. So the electron part occurs first. So the electrons are going to basically be creating that gradient. And they're being pulled across the membrane by that oxygen that's accepting them at the end to form water. Now we have this hydrogen ion concentration on the inner membrane space, and so that's going to be pumped through this ATP synthase enzyme to give enough energy to form the ATP in the mitochondrial matrix. Okay, so that's quite complicated, right? Well, let's go ahead and make sure we're looking at the inputs and the outputs of each stage one more time. So here's the first stage, glycolysis. So what goes in? Glucose. What comes out? 2 pyruvate, 2 ATP, and 2 NADH. Okay? Is what we normally. Sometimes it produces other things, but we're going to focus on the NADH. Okay, so we've brought in glucose. We've gone through that energy investment phase and then the energy payoff phase until finally we have 2 pyruvates, 2 ATP, and 2 NADH. Okay, so this is all occurring where in the cell? The cytosol or cytoplasm. Now we got those pyruvates. They're going to undergo pyruvate oxidation to form 2 acetyl-CoAs. So in that process of pyruvate oxidation, they're going to lose the electrons, so we get 2 ATP from that process, or 2 NADH from that process. Now we have the 2 acetyl-CoA. The acetic acid will enter the citric acid cycle, go through its little cycle twice because there's 2 of them, okay, per glucose. We're going to end up with 2 ATP, 6 NADH, and 2 FADH2s, okay? So far we've gained 4 total from glycolysis plus citric acid cycle. We've gained 2 NADH, 2 NADH, 6 NADH, so that would be 10 NADHs, and we've gained 2 FADH2 for 1 glucose molecule. So now all those electron carriers will migrate into the mitochondria, inner mitochondrial space, and further get used in oxidative phosphorylation. So we end up with that electron transport. NADH and FADH2 get reduced, and then all of the electron transport chain and the ATP synthase are all working to produce anywhere from 26 to 28 ATP as a general number, varies, okay? So if we add the 26 or 28 ATP from just this last step with the 2 gained from the glycolysis stage and the other 2 gained from the citric acid cycle stage, then we end up with about 30 to 32. You'll notice some textbooks have a little bit more, some textbooks have a little bit less. So this is on average, so ignore this maximum, just say it's on average. You'll see various numbers. But it's, you know, around 30, a little bit more than 30. Sometimes you'll see a little bit less than 30. It's not 2. It's going to be in the 30s or a little bit less. All right. So we're gaining lots of ATP. So here's where everything, this is a good figure to flag in your textbook. So you can see each of the stages that are occurring and then where the ATP is coming from. So you can see most of it is not coming from the first part and the second part. It's all coming from the third part. Okay, but the first and second part, what are they doing? They're getting those electron carriers ready so that we can get that maximum ATP out of it in the third stage. All right. So let's bring it all together with a little bit of entertainment by watching our Amoeba Sisters video.
Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are. Prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when the bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis, and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules, and 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now an intermediate step occurs. The 2-pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2-pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain in chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes in electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes ATP. So, in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with 2 hydrogens, you get H2O, water. If you remember from our equation, water is a listed product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other 2 previous steps. How much?
So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about, how many protons were pumped across the mitochondrial membrane. You can see more variables discussed in the factual references, and those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain in chemiosmosis step alone. Then, if you add the other two steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you can estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this was just one way of creating ATP, but like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Nuba Sisters, and we remind you to stay curious.
All right. Okay, so again, with this video, I think we're pretty much done, right, with this lecture, yeah. Okay, so what I want you to do now is, if you're still having trouble grasping the concepts, go ahead and watch the videos again. So there was that video at the beginning of the lecture and then the Amoeba Sisters video at the end. And keep writing down the chart, okay? So look at the inputs into glycolysis, figure out the outputs. Figure out the inputs for the Krebs cycle, citric acid cycle, figure out the outputs. And keep doing that until finally you can see where every process is taking place and what the results of every process is, okay? So it will take a little bit of time, but it'll eventually click in there, okay? So we'll go ahead and stop here for now. Next time we're going to take a look at that fermentation that the video talked about at the very end, as well as some other metabolic pathways, and we'll even get into a little bit of cellular division. So I'll see you next time.